So this talk is based on Eugen's recent review on machine learning in proteomics. So if you want more information, please read it. It's very informative. And I will start with briefly restating some concepts that you have already learned using this strangely familiar picture. So shotgun proteomics or bottom-up proteomics is a technique to identify and quantify proteins in the samples of interest. And in principle, this process requires two stages. First, we digest proteins into peptides using proteases, and then these peptides are fragmented inside the mass spectrometer. Then we get fermentation spectra. Fermentation spectra are looking like the ones on the right. And here we have two different acquisition methods, DDA and DIA, and the fermentation spectra for them are looking differently. So from this spectra, we have to identify our peptides and then proteins. It can be done using several methods. One of them is called database search. And it relies on our knowledge of peptides because they have oligomeric structure. And we know that bond breakage occurs primarily in the backbone. So so we can exploit fermentation spectra to identify the precursor sequence. And moreover, if we know the fermentation method, then the fragment masses can be easily calculated from the peptide sequence. We have this theoretical spectra, but as you can see, all peaks here have the same height because we don't know the intensities. Then we compare the theoretical spectra and the experimental ones using some kind of scoring engine, for example, androgynous database search from MaxQuant, and identify our peptides. But although this approach has been successfully applied for many years, intensity pattern carries crucial information that we don't just want to lose. And one way to utilize it is to assemble libraries directly from previously measured spectra. And this approach has advantage in that it's we don't rely on our assumptions about the peaks from the peptide sequences. We use the peaks themselves, so it's hypothesis free. However, any new peptides not present in the spectral libraries will be lost in the analysis, which is not good. So an alternative is the dedicated acquisition of project-specific libraries, and it solves the previous issue, but it adds measurement effort in our project. So we need to spend more money and more instrument time and so on. So measured libraries are used only when the benefits of increased sensitivity strongly outweighs the disadvantages of spending more resources. One example of such thing is DIA proteomics, because in DIA, due to many co-fermentations, the additional information from spectral libraries is particularly beneficial for untackling the contributions from different peptides. But there is also a third way, generating artificial predicted libraries, and thus we come to spectrum prediction. So what is spectrum prediction? Here we mean reconstructing the peptide fermentation pattern from its sequence. And the people have been trying to do it for nearly 20 years, but only recently modern machine learning techniques enabled nearly experimental accuracy of predictions. And this lecture will be about these techniques and the applications of machine learning in proteomics. But first, I will briefly talk about the core machine learning concepts. So what is machine learning? Machine learning is a set of methods for building models from experimental data. And there are numerous machine learning algorithms, and they can be grouped into supervised, where we have categorical or numerical values to predict, and unsupervised, where the goal is to find patterns in the data itself using clustering or dimensionality reduction. Supervised methods can be further divided into classification with the categorical targets and regression where the outputs are numerical. And spectrum prediction is the regression task where the output variables are fragment intensities. There is also deep learning. Deep learning is a particularly hot and advanced area in machine learning, which uses various neural networks as models. Neural networks are systems of connected units called neurons, which organized in layers. So the term deep in deep learning stands for the multiple layers in the network. And here we have the simplest neural network architecture. It's called multi-layer perceptron. Neurons can transmit signals modulated by weights assigned to the connections. Additionally, an activation function is applied to introduce nonlinearity to transformation. Weights can be adjusted in the process called backpropagation, and this way, neural network adjusts data and learns it. One of the hallmarks and the main advantage of deep learning is the ability of neural networks to learn higher level concepts from the data itself without additional feature engineering. And this greatly simplifies the pre-processing of the data, but comes at the cost of the need for increased quantities of data to train models and a lot of computational resources. So many advanced neural network architectures have been developed tailored for specific tasks. Among the most popular ones are convolutional neural networks, recurrent neural networks, and transformers. And all of them have been successfully applied to spectrum prediction. However, particular success have been achieved with recurrent neural networks. Recurrent neural networks, or RNNs, are designed to process sequential data, and they can be applied to sequences of variable length, which makes them particularly useful for processing of peptides. And classic RNN models had troubles dealing with information spread across long distances in the sequence, which is actually important for peptides. But modern, more advanced models, such as long short-term memory and gated recurrent units, address this issue. Now back to spectrum prediction. It can actually be done in two ways, predicting ion series intensities or full spectrum. The first and the most popular one is focusing on predefined ion series. One needs annotated spectra to train such model, and it will predict only ion types that are present in this annotated spectra. But this approach is robust to co-fragmentations. Several neural network-based models have been developed for this task. And here we have a simplified scheme of deep mass prism, which is RNN-based. It has several long short-term memory layers, which receive peptides of various lengths and transform them into fixed length representations. These representations are later sent to multi-layer perceptron, which outputs ion intensities. Conventional machine learning can also be applied, but it has to somehow deal with the variability of peptide lengths. And as Jürgen said, there are two ways to deal with it. One of them is to just build models for each of the lengths separately. This approach is, of course, the simplest one, but it loses the synergy from peptides of different lengths. So for ease of the comparison, here on this illustration, multi-layer perceptron is used as well as the model. But in principle, any machine learning model can be used here. This approach is implemented in MS2PIP, which uses XGBoost. The second way is window-based approach. Here we have the sliding window centered on peptide bond, and since the window itself has fixed length, it transforms our feature space from variable length to fixed length one. And then we can use different models as well, multi-layer perceptron in this case. And this approach is implemented in Vina. An alternative is predicting the full spectrum as is without referring to ion annotations, ion series annotations. And this approach is complicated by cofragmentations, so they need to somehow be, reduced, for example, by spectrum clustering. But the advantage of this approach is that it can predict non-backbone ions. The first and the only model utilizing this approach was developed in 2020 by Liu et al. So now let's look at some projects that applied intensity information to improve peptide identifications. First, let's look at DDA projects. And in DDA, intensity information can be integrated into search engine score to increase sensitivity, specificity, or both. For example, in this work, HeLa wholesale lysate was analyzed with MaxQuant. Then undropping the search score was augmented with predictions from 3 different models: DeepMass Prism, Vina, and MS2PIP. So as you can see, the improvement in sensitivity is Q-value dependent and is higher at small Q values, which is good because this is the area that we are most interested in. And here we have the same plot, but zoomed in to the range from 0 to 1%. And this is the gain in PSMs. And here the gain in peptides. So at a standard cutoff of 1% FDR, the improvement with the best model was around 4%, which is good, but not spectacular. But one can expect that in the larger search spaces, the benefits will be larger because there we have, on average, more PSMs in a given tolerance window from which we need to find the correct one. And we have such spaces in immunopeptidomics and proteogenomics. So immunopeptidomics focuses on peptides derived from human leukocyte antigens, or HLA for short. And these peptides are generated by proteasomal cleavage inside the cell, then relocated to the cell surface and presented to immune cells. They are quite small, so they can be directly measured by mass spectrometry. However, the cleavage is unspecific, so they have no information about terminal amino acids. And so we have the aforementioned larger search space. In addition, the rules of fragmentation for non-tryptic peptides are different. So models have to be trained specifically on non-tryptic peptides. But a trained model can yield really good results. For example, in this study, Wilhelm et al. Trained the PROSIT model and reprocessed with it a data set consisting of HLA peptides from 95 different monoallelic cell lines. And they rescored with the model proposed PSMs. As you can see, here the green bars represent the quantities of peptides gained, blue bars peptides shared, and red bars peptides lost compared to the data set before preprocessing. And overall, we were able to achieve on average a striking 1.5 improvement, a 1.5-fold improvement in terms of identified peptides. So yeah, that's pretty good. Similar scoring studies also suggest that integration of intensity information is clearly beneficial for the analysis of HLA ligands. Another field that can benefit from this integration is proteogenomics. It's the study of the proteome with the aid of genomics on transcriptomic sequences. And this way, it can identify peptides that are not present in reference proteome. but this requires in silico translation of various degrees from the inclusion of untranslated regions of transcripts, so 3' or 5' UTRs, to the 6-frame translation of the whole genome. And Verbruggen et al. Have shown that proteogenomics can also benefit from the integration of predicted spectral intensities. They generated proteogenomic search spaces using ribosomal profiling and the free-frame translation database based on RNA-seq. Then they used MS2PEEP and PROSIT to augment Andromeda search scores. So similar to the standard DDA analysis, the improvement is FDR dependent and is larger at highly specific regions. And at the standard cutoff of 1% FDR, the increase in identifications was around 6%. Now, if we compare the benefits that spectral predictions provide to immunopeptidomics and proteogenomics, one can see that former is a clear winner. And this can be somehow related to the fact that it's dealing with non-tryptic peptides. But then we move to DIA, and as I've already told you, DIA increased sensitivity from spectral libraries is particularly important. So many DIA analysis workflows directly utilize measured spectral libraries to query samples. So in principle, they can use, instead of measured spectral libraries, predicted spectral libraries. And it was shown that it is advantageous, at least for the standard DIA samples. For example, here we have principal component analysis results of tissue samples of different cancers. And the data on the right was measured with, was analyzed using cancer type specific measured libraries. And on the right, the same data was analyzed using MaxDIA with predicted library of all human tryptic peptides, including up to 1 missed cleavage. And as you can see, it resulted in more protein groups and better separation of tumor types in the principal component analysis plot. What is also interesting, and Eugen briefly mentioned it, but I will repeat it because it's important, is how different predictors affect the results of the identifications. So here, the same DIA analysis on human samples was performed with spectral libraries from different models, DeepMass Prism, PROSIT, and VINA. And as you can see, there is a huge overlap in both the number of peptides and genes. So why is it important? It's because it implies that simpler and hence faster VINA model can be used instead of RNN-based ones without significant drawbacks. And since VINA is faster and requires less training data, it's ideal for creating project-specific prediction models that are adjusted for specific experimental conditions. So, for example, it adjusts to non-standard proteases, non-tryptic peptides, non-standard fermentation energies, and so on. So it can be really useful, and for that reason, we started work to make VINA available to the community. But in the process of this work, the VINA has changed. So as I've told you, any machine learning model can be used instead of multi-layer perceptron here. So we replaced it with XGBoost, and this way we were able to improve prediction quality and speed up the process even more. So we changed the name to reflect this. We have now Xmas instead of VINA, So X stands for XGBoost, mass stands for mass spectrometry, and Xmas stands for some kind of Christmas that was lost in time. So Xmas is accessed through REST API and can be operated in two ways. The first is the public mode, in which there is a public server that holds curated models that can be queried for the predictions. And you can access this server using this web address, but unfortunately, you can't train your own models there. But on this address, you can also find the GitHub link to the repository holding the source code of Xmas. And using it, you can launch it in a private mode. So the private mode allows to train models locally and then use them for predictions and potentially upload them later into the public server for the community. So when you launch Xmas locally, it produces a new tab in the browser, and there you can specify the folder or hold these MSMS TXT files from MaxQuant. You can hold one such file or several. Xmas will merge them, and then you set score threshold, and you can create models for specific fragmentation methods and charges. Xmas can be used not only for intensity prediction, but also for intention time prediction, which is done using recurrent neural networks, so it will be slower. And finally, I will briefly talk about the special cases for intensity prediction. So first is post-translational modifications. So it's covalent modifications of the peptides, as you know, and they can give rise to additional fragments, and they change the masses and the intensities of the ion series members. So several models can be adapted to predict PTM data, for example, PDIP2 and MS2PIP. Also, there are specialized solutions such as deep phospho. The second special case is cross-linked peptides. There, a special linker is used to covalently connect two peptides, or sometimes more, and this significantly complexes fragmentation spectra and makes predictions harder. Furthermore, we just have less data available for cross-linked peptides, and this data is more diverse due to the existence of different linkers. But there is one RNN-based model for predicting cross-linked peptides, which is called PDIP-XL. So that's it. Thank you for your attention, and I will be glad to ask your questions.
Hello. Sorry if I misunderstood something, but I mean, you showed that the in silico libraries outperform like experimentally derived libraries, right? In terms of numbers that you got in the end.
Yes.
Like for example here, yeah?
Mm-hmm.
But I was actually wondering how, I mean, like how big are these two libraries? I mean, do you start with the same number of like spectra in both of the libraries? Like if you would compare like these numbers in percentage, like, because I mean, you always depend on the size of the experimentally derived library, right? Mm-hmm. And then if the size of the experimentally derived library is the same as the in silico one, or in silico one is just bigger and that's why you got higher numbers?
Yeah, so the in silico database will be much bigger. So first of all, I mean, there's the factor that this has all uniprot proteins, while the experimental library will only have, let's say, half of the proteins or something like this, if it's really good. But also the sequence coverage, it's complete in the in silico prediction, and it's 20–30% in the measured library, right? So you can say could be around a factor of 10 larger, but it's not, the argument is not like this, that because it's larger, you get more. Because here you have the same problem as with DDA search spaces. If you have a larger search space, you also generate more false positives. So it's not trivial that just because the library gets bigger, you have more identifications. Maybe that was like this in the old days when the DIA softwares couldn't do good FDR control, but now with the proper FDR control in the DIA softwares, this is a non-trivial thing, that by offering larger library, you also have more DIA identifications.
Okay, but would you say that there is still a chance that experimentally derived library can outperform at any time in silico library?
Yes,
I mean,
yes,
okay, thank you.
Which it does. So in some instances for timsTOF data, we see this actually happening.
Thank you.